Welcome to Parasitology at Leiden University Medical Center. I'm Maria Yazdambach and heading the department where scientists and clinicians are working together towards the development of vaccines against the most devastating parasitic infections. We are also learning from parasites how to modulate the immune system or our metabolism, as we believe this can lead to novel treatments for allergies and type 2 diabetes. Regarding vaccines, our malaria research is at the forefront of malaria vaccine development. The genetic modification of malaria parasites was pioneered at our department. This has culminated to the development of the first genetically attenuated malaria vaccine. This is now being produced by Sanaria, a US-based company, and is being tested in human volunteers in the Netherlands by us and by our collaborators. Malaria researchers continue to improve the genetic modification of malaria parasites, now using CRISPR-Cas9 technology to create better vaccines. We're also contributing to the development of vaccines against schistosomiasis and soil-transmitted helminths through working in large international consortia. What sets us apart is our focus on the glycobiology of these helminths. Through our glycomics program and the glycoprotein arrays, we are identifying new molecules that are the target of effective immune responses for vaccine development. When it comes to understanding how parasites manipulate their host, there are data showing that helminths can actually have beneficial effects on asthma or type 2 diabetes. Pinpointing how exactly this happens and which parasite-derived molecules, particularly glycans, are responsible for the manipulation of the immune system or modulation of metabolic homeostasis can provide us with unique therapeutic strategies for allergies or for controlling insulin sensitivity and metabolic syndrome. Our laboratory investigations are linked to strong field studies within the framework of long-term collaboration with institutions in Africa and in Southeast Asia. Training and capacity building has created equal partnerships where we learn from each other. And it also helps our translational and clinical research to reach places where they are needed. Indeed, in an academic setting, research is always linked to teaching. We train our students and our colleagues not only in classical parasitology, but also in a wide range of topics such as immunology, epidemiology, and public health. Parasitological research cannot be separated from strong diagnostic research, and our department over the years has been very committed to this. We've developed a number of sensitive and specific molecular diagnostic procedures that are currently being used in many parts of the world. Our circulating antigen tests for schistosomiasis are currently the most sensitive field-applicable diagnostic tools available for mapping schistosomiasis. These are becoming the tests of choice for large-scale control and elimination programs. Continuous innovation is essential. In this respect, one of the most exciting recent steps taken has been the creation of the Leiden Center for Controlled Human Infections, where volunteers are infected with a pathogen in a controlled setting with utmost care to ensure absolute safety. Such controlled human infection models have transformed the field of vaccine or drug development. Is a vaccine or drug candidate effective? To answer this question, this has to be tested in the field and can take years to get an answer to. Whereas with controlled human infection models, we can have the answer within weeks or months. This, importantly, saves costs and time. Another important aspect of controlled human infections is their value in telling us how parasites modulate their host, both immunologically and metabolically. Innovation in our technological platforms at the LUMC are allowing us to take systems biology approaches to the problems we are tackling. Our flow cytometry facility and our state-of-the-art mass cytometers is allowing us to characterize the immune response at an unprecedented detail. We use cutting-edge data processing programs to map and visualize the induction, expansion, and dynamic changes during controlled human infections before and after vaccination. This facilitates the understanding of immunity and discovery of correlates of protection. These approaches applied to non-endemic and endemic populations are giving us interesting insights into, for example, why some vaccines are less effective in endemic areas or in pre-exposed individuals. We also have experts in the area of immune metabolism. The skewing of the immune system through alteration of the cellular metabolic pathways is providing new avenues for specific and targeted changes of immune cells, with implications beyond vaccine development and beyond immune modulation. Our approach to go from basic into translational, followed by clinical and epidemiological population research, showcases how fascinating and how multifaceted parasitology can be.